Neil Hall from the Earlham Institute. Neil?
Hi. Yeah, I'm just sharing my screen.
Yeah, so please start.
Okay. Can you see that?
Yes.
Okay, right. Thank you very much for inviting me to speak at this meeting, particularly to thank God for all the effort of putting this together. So I'm going to talk about a little bit about some work that's gone on in my lab at the Earlham Institute on the Euglena gracilis genome, but also put in the context of the larger projects, one of which you heard about today, the Earth Biogenome Project, and then the Darwin Tree of Life Project. And I'll talk about the component that on protists. So Mark Blackster will be speaking right after me, and I'll probably go into much more detail about the project at large. So firstly, I should say that what we do is data-driven science, and in the biosciences, it kind of gets a bad press. So if you think of data science as the field that uses methods, processes, algorithms to extract knowledge from structured and unstructured data, it's very commonly used in the physical sciences. There's the Large Hadron Collider, which, although was set up initially to test the hypothesis of the Higgs boson, once it did that, they just started banging particles together to see what comes out. I'm sure it's more complicated than that, but that was, for the most part, what they had been doing. Then you have things like the Hubble telescope, which looks into space, looks for new structures and stars and planets, etc. When the biological sciences, we often label anything which is data-driven as stamp collecting. In fact, well, people who review my grants tend to do that. However, there is a good history of data-driven science in the biosciences. The human genome was data-driven science. Darwin's Voyage of the Beetle was data-driven science. He went out, collected data without a starting hypothesis. And you heard from Harris previously about the Earth Biogenome Project, which aims to sequence all of the described species on Earth, as well as collecting data on new species, and is anticipated to collect over an exabyte of data. And to put that into scale, all printed material on Earth comes to 0.2 exabytes. So it's a huge amount of data, and something which is likely to transform biology. And what at the Earlham Institute and my group have been involved in is the Darwin Tree of Life project, which is a sub-project, if you like, of the larger network of the Earth Biogenome Project. The DTR consortium made sequence all species in Britain and Ireland. That's meant to say Ireland, not Iceland. That's a mistake. Over the next 10 years, led by the Sanger Institute and Mark, who's speaking after this, and all of the partners that you see there, which includes everything from academic organizations to collections organizations and universities, to really collect and gather all of the species and process them for sequencing. And at the moment we have initial pilot funding, which we hope will be extended. Just to echo what Darwin's original note on the Tree of Life there, when the consortium came together on the 4th of May 2018, everyone signed this document to say we are going to sequence all life in the British Isles and make it freely available to the world. And that's the kind of the plan that's gone forward. And you'll notice that Mark Blackster is one of the key signatures on there, which is just as well, because he has a responsibility of doing that. Now, unfortunately, for this community, that although there's a huge amount of expertise in genomics that's been gathered over the past couple of decades, the majority of the scientific community are really interested in this branch of the Tree of Life. And this is one of Sandy Baldwin's original Tree of Life. But some are interested in fungi, and there's others interested in plants. But really much most of the Tree of Life is kind of neglected. So 99% of the community are working on 1% of the diversity on Earth. And so what we're hoping to do is try and add some genomes to the rest of this tree, including the organisms that the people on this meeting are interested in the discobar, the Euglenids. And mainly because they're hugely interesting. The protists are hugely interesting and beautiful life forms, but also hugely diverse. And if you can understand all of the different mechanisms and processes which enable these forms of life to exist on Earth, you kind of identify all the different solutions to life there is, and we don't want to limit ourselves to such a small amount of diversity. And so if the aim of these projects is to describe life, they really need to describe all of life in this tree, not just the life that we can see. So in the Darwin Tree of Life, we've been driven very much by species lists, and those species which exist in culture collections. So the major culture collection in the UK is CCAP, and they have a list here of all of the different cultures in the clade of Euglena. And we started by getting some of these in for culturing, and which will be then sequenced at the Sanger Institute to produce what we hope will be highly contiguous draft genomes, but that will be complicated, though no doubt Mark will speak after me and talk about some of the advances that have gone in technology, which have meant that it can go from DNA rapidly through to very high quality genomes with nearly telomere to telomere contiguity. But one of the problems facing people working on proteins is a large number of them can't actually be cultured, and therefore you can't produce large quantities of DNA at a high molecular weight to be able to produce contiguous genomes. So at the Earlham, what we're piloting is a different approach, going from single cell sequencing and applying something we call GNT-seq, which is genome and transcriptome sequencing, which enables you to go from an individual cell, extract both the DNA and the RNA for sequencing simultaneously. And so we're going from water samples and then from bulk samples generating community DNA for long read sequencing and potentially high-C sequencing. And then from the same sample, using cell sorting to sort into 96 or 38 well plates, the different organisms from the community, then extracting the genome and transcriptome, and then combining the data from the single cell data to long reads so that we can bin the metagenomes as outlined in this picture. And we've started to do this, and I don't have time to go through the preliminary data today, but we have been able to generate some reasonable genomes with, directly from single cells, not yet incorporating the long read sequencing, and getting, over 60% BUSCO scores from those. Although, as you'll know from environmental samples, you also get a lot of contamination with you. So there's a lot of development we've got to do around the bioinformatics to generate really high quality data from this. And then we can then also use the transcriptomes to allow us to add annotation to this. So that's where we are with the Tree of Life, and I will then now go on to talk a little bit about the work we've done on Euglena gracilis, and this has been done by Ross, who's at this meeting.
Neil,
yes.
Neil, please hurry up.
Yeah. Okay. And it gets there. Just to, how much over am I? Okay, so I'll just say we've generated paired end. We've got some make paired and paired end data from the community previously, and we've started trying to incorporate 10x data, which is from single molecule, going from single molecules of DNA to be able to generate data from essentially haplotypes. And using a supernova assembly to try and incorporate this into the data that Thank God used in his original paper. And one of the first things that we identified from this using KMA analysis is that you see, if people aren't familiar with cat plots, this is looking at KMA, the coverage of KMAs and the number of KMAs you see in assembly, and using both the 10x data and just the normal paired end data, you see 3 peaks here, which is usually indicative of a triploid genome rather than a diploid genome. So it may well be that one of the reasons that we're having so much difficulty with this assembly is it's not a normal diploid as you might expect. And this gives you some of the assembly statistics. We have been able to slightly improve the N50 that, thank God, originally produced using our wrap assembler to 2 kb, but you can see there's large discrepancies in the genome sizes. So as I'm over time, just to say that the Euglena community was still waiting for a high quality genome and will probably keep waiting for a while. But we need to new methods to generate high quality drafts, and hopefully Mark talks about some of those next. And hopefully the projects such as DTOL will be generating many more of these genomes over the next couple of years. And we'll hopefully be able to learn a lot from this environmental single cell sequencing to identify new species potentially and generate genome resources. And I think one of the main things for this meeting is the community need to pool their effort and expertise to make the most of these resources. Thank you, and I just want to thank all of these people that have been involved in this.
Thank you, Neil, for introducing your very ambitious project. And I think we can continue in writing down your questions into the chat,